Good evening to everyone who is just listening to this particular lesson on translation. You know, translation is a very complicated process that happens in all the living cells, whether it is a prokaryote, eukaryote, plant, any living being will have to perform translation and get the proteins. Now, in the most simple way, what we can say would be the 4 base pair letters ACGU of ribonucleic acid. There are 4 letters that will be converted into 20 amino acid letters into polypeptide chain, that is protein. And that happens in the cytosol. That means in the cytoplasm. So in the cytoplasm, the 4 base sequence of ribosome nucleide will be converted into a polypeptide chain, and that process is generally called protein synthesis, and that takes place in 3 important steps. Now, before that, what we should know, in the cytosol, there will be the mRNA and there will be tRNA produced by the nucleolus, and there will be many more macro molecules, and there will be ribosomes, and all these macromolecules will have an interaction. They interact, and then with the help of enzymes, there will be more than 25 enzymes that would take part in the protein synthesis. And also there will be factors like protein factors, and there will be energy molecules like ATP and GTP, and then there will be 20 amino acids. They are the major players. So 20 amino acids, the mRNA, tRNA, ribosomes, and along with this a large number of chemical macromolecules. They will have a large amount of interplay, and complicated interactions will happen. All of them will happen in fraction of a second maybe, and they will form a long polypeptide chain called the protein. You should know that the human body, in any cell for that matter, one gene can produce 10 proteins. One gene can synthesize 10 different kinds of proteins. It has been estimated that in the human body, when you take the human body, it is said that there may be 80,000 to 4 million protein types. Isn't it wonderful? Can't we feel how kind of a idea, because a single cell in the living cell in the human body or in any living being can produce such wonderful chemicals like macromolecules. So this would be the thing. We require some background information because most of them are all happening in the cell, and they are all biochemical reactions and organic reactions. We shall try to know a few of them. One of them is called the peptide link. That you should know. Peptide link between the amino acids. When 2 amino acids want to join together to form a chemical bond, then you get the peptide bond. If you want to know what it is, now this is one amino acid. You know, amino acid will contain one carbon compound called alpha carbon. To that you have 1, 2, 3, 4 groups of molecules will be attached. One of them will be hydrogen, and the important one is the NH2 or amino group, and there will be one called R1 group, that is the side chain. It may contain any organic molecule, and then we have a carboxyl group containing COOH. So now this is another amino acid taken. Now when these two amino acids come together due to attraction, there will be a peptide linkage. What happens is the hydrogen of the carboxyl group of one amino acid will join with the hydroxyl group of another one, and that will be water, that is hydrolysis. Now that water will be removed, that will go away. Then what we left is a carbon, one amino acid, second amino acid, and a link between them. That link will be called peptide bond. I think you should understand that particular point. Peptide bond happens between two amino acids by removal of water, and then at the carboxyl group and the amino group, they join together and they form what's called a peptide link. So we say that protein is a polypeptide chain. A protein may contain thousands of amino acids linked together by means of polypeptide chain. So you should be able to remember that. Now from there we come to another important thing. There are 20 amino acids available in the cell pool. Inside the cell we take food. The food will be converted into different molecules. One of them will be amino acids. So amino acids will have to come from outside, and there will be a pool of amino acids in all the living cells, and whatever we find, we only have 20 amino acids. From these 20 amino acids, there may be about 80,000 or more proteins by different kinds of modifications. We shall look into that little later on. So if you understand this particular thing, saying that protein synthesis is a very complicated process, in that four important players are: one is mRNA, tRNA, ribosomes, and amino acids, plus energy molecules ATP and GTP. These molecules will take part in the formation of the protein synthesis. Now I said there are three important steps in the formation of a polypeptide chain. One of them is called the initiation, second is called elongation, and third is called termination. When once a polypeptide chain is released from the site of its manufacturing, that is in the rough endoplasmic reticulum, RER, that will contain the ribosome, and the ribosome that will be modified into various kinds of proteins. So to begin with, we shall know what is the first thing called initiation. Now initiation happens on this mRNA molecule. You know the mRNA molecule, it has the 5' end and 3' end. Now this mRNA molecule will be there. For that, initiation factors, that will be some proteins, enzymes will join together, and they will bind here, and they will make the smaller subunit. This is the smaller subunit of the ribosome. So smaller subunit that will combine with the mRNA molecule. Then at the same time, there will be another one, the larger part of the ribosome, larger unit, and in this larger unit there will be some enzymes, they take part, and to that one we get a tRNA which is acylated, that will come and that will deposit. So this entire structure will become what's called the initiation complex. That is the smaller subunit of the ribosome first get attached to the mRNA with the help of a binder molecule or a factor. Then the larger unit of the ribosome will collect one tRNA that is acylated, we will explain what is that, and that will come with the methionine part, and they will get into the first part of that 3-letter codon called the initiator codon or start codon, that will be AUG. So that will come and attach, and all this we call it initiation. That means the mRNA containing all the codons, they are called the codons, 3 letters of this ACGU. What I do, I will just try to remove this part so that we will be in a position to understand that slightly better. I will remove this portion now, and we shall try to find out quickly what is that we are talking about. Now we have A adenosine phosphate, A, then C cytosine, then guanine, and then uracil. These are the nucleotides on the RNA. Now 3 of them will form a codon. So you have 1, 2, 3, 4 of them, and when they start combining into forming, they become 4 times raised to the 4 raised to the cube 3, that will become 64 3 letters. That if it is one thing, then it will be ACU, then it may be UGC, like that. There will be 64 codons will be available on the mRNA. Each of the 3 letters is known as one codon. So the codon will be there, and 3 of the codons will be not responsible for any amino acids. One of them, AUG, the first one, this AUG is called the starting codon. So out of the 64, one of them will be starting codon for any initiation activity on the mRNA. AUG must be there. So in the initiation complex, the first codon will be AUG on the mRNA. Then 3 of them, like you go away, UAA, like that, UGA, UAA, and one more. These are called the stop codons or stop signals. So 3 of them will be stop signals, and one of them will be starting codon. So now we understood. Now there will be 61 codons available for the translation. Now then we should just, this also we will remove it now so that we can go to the next part of it. Now this tRNA is there. In the tRNA, if you look at the tRNA, it has got a clover leaf model like structure, and it has got a structure that is almost like that. And this will contain inside the codons, the nucleotides, nucleotides will be there. And this is called the tRNA, transfer RNA. This will contain here one part. If you look at the parts, now this is called the this one arm called DRM. This is another arm called TRM, and this is the anticodon end, and these three will form the anticodons. Now this arm tries to identify the amino acid, and it has got a 5' end and a 3' end, and in this 3' end we have C, A, that is the nucleotide, adenine nucleotide, cytosine nucleotide, and cytosine nucleotide, that is N. That is the structure of the tRNA. Now this tRNA molecule will have to bring amino acid. It can pick up an amino acid. Now, how does it pick up? For example, if you take the first one called methionine, that is the first codon, AUG is fixed already. For that, there must be UAC anticodon, and that will be, supposing on this tRNA, you have U, then for A, for U you get A, and then for G you get C. If this is the anticodon for the first codon of the mRNA, to this methionine will have to be attached at the A adenine site. And here what happens is, with the OH group of this one, this will get attached, and that is called amino acylation, or you can say acyl amino acid. It comes. For that purpose, ATP is used, and ATP will give the energy for that. One of the things will go, and this type of that is the tRNA molecule, where the amino acid is added. That is called a charged tRNA. So a charged tRNA comes here, this one. Now when this site, if you look at it, it has got one A site, P site, and E site. A site means arrival site, or it is called amino acid site, and P is called peptide site, and E is called exit site. Now when once this has happened, the entire structure, that is the ribosome lower part, upper part, containing the AUG and UAC, then they move towards 3' end, that is reading. Reading takes place. It moves one place away, and now it will sit on this particular thing. And then for AGA is arginine. Now that means initiation is over. Let me stop here. In the initiation process, the charged tRNA comes and occupies a place on the initiator codon. Initiator codon is AUG. To match that, there will be a tRNA molecule carrying methionine amino acid, and the anticodon will be UAC. So when on the P site, it's called the peptide site, on the P site, when the methionine tRNA comes and matches with the AUG, at that time the initiation is over. Therefore, all the factors which are enzymes which are associated with the initiation complex, they will all go away. Now this is ready for the next step called the elongation. I hope you are clear now by this time that the initiation is a process where the two subunits of the ribosome will come together and they get attached to mRNA molecule. At the same time, on the initiator codon on the mRNA is always AUG. To match that one, there will be a tRNA a charged tRNA comes with UAC as the anticodon and methionine as the first amino acid, and this will be the initiation. The next step will be the elongation. In the elongation, when the elongation happens, the whole complex moves forward. When it moves forward, what happens? Now, what happens is that one of the next reading comes here. This contains AGA, arginine. That is the codon for arginine. So the tRNA or the two, any one of them can come in, but to match that one, this comes inside and deposits here. That will be U, G, then U, and that will be the tRNA carrying the arginine. Comes and takes the A site. That's the A site. So when once that enters into that one, that means there will be two different amino acids will be taking a position, one on the A site, one on the P site called the peptide site. Now when once they come inside nearby, attractions will happen and the biochemical activity will go on, and there will be peptide linkage. As I told you, there will be peptide linkage. When once the peptide linkage happens, then this particular amino acid get attached to A site now. So that is the first thing that happens, A site. Now as this moves now to the next one, let's say it moves to the next one. Now I will remove this and show that now it has just moved slightly one step away like that. It has moved. When it has moved, now this is now P site is almost empty. Our factors are gone, and the P site, now this entire thing moves again one step forward. As it moves, this area becomes empty. It has gone, and this is the exit site. It comes and this goes away. So now what is left will be, this also goes away. What is left will be in the A site, and now that moves to, this moves to P site. As it moves to P site, now this will contain U, G, U. This has gone, and now this will have 1, 2 amino acid. It has moved now. So 2 amino acids are there. Now next one comes here and that attaches to this one, and let's say that attaches to the other codon. And then this P site, in the A site again it comes, and it comes with a amino acid, and these two will get linked by peptide linkage, and then this goes away. You get now, this will be shifting to A site. You get 1, 2, 3 amino acids. Like that, this keeps on going. That's what's called the elongation. And I will stop here for some time and see what will happen next.